Hi, my name is Christian Rebolletto, and I will be presenting the effects of hypothalamic stem cell degradation on systemic aging. Now, before we begin, it's important to understand two things. One, what stem cells are and how exactly they function, and two, what aging is and how it is a disease. Now, the first of that is, of course, how stem cells work and what they are. Stem cells are essentially cells that are able, through pluripotency, to become different kinds of somatic cells. So, under the right conditions, a stem cell can become a muscle cell, a nervous cell, any kind of somatic cell out there, depending upon the environment that it's placed in, which is induced pluripotency. Now, aging is often debunked as just a process that naturally occurs, which it is, but it's also a disease and it's not often looked at as such because a disease essentially causes biological degradation in function, and that's what aging does through the production of diseases that are age-related, such as cancer or Alzheimer's. Now, with these two ideas in mind, it's time for me to get into the actual study that I did on hypothalamic stem cell degradation in regards to mice specifically. So in this first figure here, figure 1, we can see that htNSCs, or hypothalamic neural stem cells, which they'll be referred to as htNSCs throughout this, produce what's known as exosomes. And exosomes are essentially just products such as proteins or microRNAs that are expelled from the cell. And these microRNAs and proteins are able to interact with other cells through intracellular connections and affect vast cellular organizations and tissues. Now, with this knowledge, we primarily are going to focus on the CD81 protein and the HNSCR long non-coding protein, which interacts with YB1. Now, in the second figure here, it's seen that hypothalamic stem cells, or htNSCs, are great in number and potency at 3 months old in the study done on mice. However, at 18 months old, the potency and the number of these htNSCs are vastly reduced. Now, this is due to the fact that over time, the production of long non-coding RNAs or microRNAs and proteins within the cell lead to something called cellular senescence. And cellular senescence is essentially the zombification of cells, where they do not die, but they do not perform their biological functions in the way that they're intended to. This causes a buildup of senescent cells, which overall reduce the function and of organs and organ systems, such as the brain, and causes issues like Alzheimer's or overall cardiac health being reduced. So this idea is very important because when we look at aging, the diseases that are a result of aging are often relative to the biological congestion of these senescent cells, such as Alzheimer's, which is still a very new tapped into disease, but also cancer, which is an accumulation of cancerous cells. And overall, this is brought on because you don't have a large amount of immune protection as stem cells decrease. And because the hypothalamus has such a high role in regulation of hormones and cellular function, a decrease in the stem cells maintaining cellular health within your brain and your overall body will, of course, result in a decrease in your body's health and protection, causing age-related diseases such as cancers or even just rheumatoid arthritis, other diseases relative to that. Now, this kind of concept was seen particularly in mice through the analysis of the HNSCR long non-coding RNA and its interaction with the YB1 protein. So RNA, when it interacts with certain proteins or ribosomal proteins, there is a production of microRNAs that sometimes, especially with lncRNAs or long non-coding RNAs, are expelled because they are not necessary to the function of the cell. Now, although this might not be a direct effect of lncRNAs, it does not mean that they cannot be expelled from the cell and interact with others through cellular reception or just protein binding. So because these RNAs and other kinds of molecules are being expelled from the cell, does not mean that they do not serve a function or a purpose in the relative long-term situation that is systemic aging. So over time, the HNSCR RNA is not being produced in the number that it is originally at a younger age, which causes a reduction in microRNAs that help maintain cellular health which overall causes cellular senescence in the already low numbers of htNSCs. This, of course, results in reduced biological function, which is seen in figures 3 and 4 over here. So the blue represents mice that were experimented on that did not have any reduction in artificial HNSCR, while the red represents mice and individuals with a null amount of HNSCR, long non-coding RNA. So in the first graph in figure 3, we can see that individuals with HNSCR null had about a 25% decrease in both muscle endurance and coordination, as well as other factors such as neural functionality and overall biological function. Now, this is a result of microRNAs not being produced by exosomally by htNSCs, resulting in cellular senescence, which of course in turn will result in the negative impacts that aging has. And this can be seen through the second graph in figure 4, where the HNSCR null population had a lower survival rate than those that had plentiful HNSCR populations of microRNAs. And this means that the senescence of hypothalamic stem cells in particular reduces the biological functionality of mice, which can also possibly be seen in humans as well. Because mice and people share a lot of neuromicrobiology, it makes sense for in the future to test these kinds of theories and hypotheses on human beings as well. Overall, because of the lack of HNSCR and other microRNAs and proteins such as CD81, there is a reduction in biological function through different organ systems, which overall may be able to be applied to humans as it was in mice. So because htNSCs or hypothalamic neural stem cells produce long non-coding RNAs as well as microRNAs and other proteins, the reduction in the amount of htNSCs present in the hypothalamus overall reduces the amount of proteins maintaining cellular health, which avoids cellular senescence overall. But of course, with the overaccumulation of senescent cells, especially in the hypothalamus, which regulates a lot of hormonal and neuronal pathways, will result in the negative impacts that's seen attached to systemic aging. So over time, the interactions between CD81 with other cells, as well as the extracellular interaction that the proteins produced by YB1 transcription of HNSCR, will cause the phenomena that is systemic aging and overall degradation of biological health. These findings are very important in regards to human longevity as well as stem cell research, particularly because since mice are homologous to an extent in neurobiology with people, it may be possible to, in the future, research on the effects of certain proteins and exosomes produced by htNSCs in humans and see how that affects the possible diseases throughout aging in different kinds of age groups. Additionally, it may be possible through certain findings, such as these in mice, to actually turn back the clock on the diseases attached to aging and age-related infections and issues such as biological degradation in humans, as it was done in mice through the studies seen in my references. Overall, I would like to thank Brittany for providing me support on this project, as well as UNLV and the OUR project or organization for allowing me to share this knowledge with my peers and peers that have similar interests in biological sciences. Thank you.